It's Eric Topol from Ground Truth, and I've got some really exciting stuff to talk to you about today. And it's about the announcement for a new center for pediatric CRISPR cures. And I'm delighted to introduce doctors Jennifer Doudna and Priscilla Chan. And so, first, let me say this is amazing to see this thing going forward. It's an outgrowth of a New England Journal paper and monumental report on CRISPR in May. So, let me introduce first, maybe Dr. Doudna. Jennifer is the Lee Ka-shing Chancellor's Chair and a professor in the Departments of Chemistry and of Molecular and Cell Biology at the University of California, Berkeley. She's also the subject of this book, one of my favorite books of all time, The Codebreaker. And as you know, the 2020 Nobel Prize laureate for her work in CRISPR-Cas9 genome editing. And she founded the Innovative Genomics Institute back 10 years ago. So Jennifer, welcome.
Thank you, Eric. Great to be here.
And now Dr. Priscilla Chan, who is the co-founder of the Chan-Zuckerberg Initiative that also was started back in 2015. So here we are a decade later with these two leaders. She is a pediatrician, having trained at UCSF, and is committed to the initiative, which has as its mission statement to make it possible to cure, prevent, and manage all diseases in this century. And so today we're going to talk about a step closer to that. Welcome, Priscilla.
Thank you. Thanks for having me.
All right, so I thought we'd start off by, how did you two get together? Have you known each other for over this past decade since you both got all your things going?
Yes, we have. We've known each other for a while, and of course, I've admired the progress at the CZI on fundamental science. I was an advisor very early on, and I think actually that's how we got to know each other, right, Priscilla?
Yeah, that's right. We got to know each other then, and we've been crisscrossing paths. And I personally remember the day you won the Nobel Prize. It was in the heart of the pandemic, and a lot of celebrations were happening over Zoom. And I grabbed my then 5-year-old and got onto the UCSF celebration. And I was like, look, this is happening. It was really cool for me and for my daughter.
Well, it's pretty remarkable convergence leading up to today's announcement. But I know, Priscilla, that you've been active in this rare disease space. You've had at CZI a Rare as One project. Maybe you could tell us a bit about that.
Yeah. So at CZI, we work on basic science research. And I think that often surprises people because they know that I'm a pediatrician. And so they often think, oh, you must work in healthcare or healthcare delivery. And we've actually chosen very intentionally to work in basic science research, in part because of my training as a pediatrician at UCSF. As you both know, UCSF is a tertiary quaternary care center where we see very unusual and rare cases of pediatric presentations. And it was there where I learned that how little we knew about rare diseases and diseases in general, and how powerful patients were, and that research was the pipeline for hope and for new discoveries for these families that often otherwise don't have very much access to treatments or cures. They have a PDF that maybe describes what they have, what their child has. And so I decided to invest in basic science through CZI, but always saw the power of bringing rare disease patient cohorts. One, because if you've ever met a parent of a child with rare disease, you know they are a force to be reckoned with. Two, they can make research so much better due to their insights as patients and patient advocates. And I think they close the distance between basic science and impact in patients. And so we've been working on that since 2019, and has been a passion of ours.
Wow, that's great. Now, Jennifer, this IGI that you founded a decade ago, it's doing all kinds of things that are even well beyond rare diseases. We recently spoke, I know, on Ground Truth about things as diverse as editing the gut microbiome for an asthma and potentially someday Alzheimer's. But here you were very much involved, IGI, with the baby KJ Muldoon. Maybe you could take us through this, because this is such an extraordinary advance in the whole CRISPR cures story.
Yes, Eric, it's a very exciting story, and we're very, very proud of the teamwork that went into making it possible to cure baby KJ of his very rare disease. And in brief, the story began back in August of last year when he was born with a metabolic disorder that prevented him from digesting protein. It's called a urea cycle disorder, and rare, but extremely severe, and to the point where he was in the ICU and facing a very, very difficult prognosis. And so, fortunately, his clinical team at Children's Hospital of Philadelphia reached out to Fyodor Urnov, who is the director of translational medicine at the IGI here in the Bay Area. They teamed up and realized that they could quickly diagnose that child because we had an IRB approved here at the IGI that allowed us to collect patient samples and do diagnosis. So that was done. We created an off-the-shelf CRISPR therapy that would be targeted to the exact mutation that caused that young boy's disease. And then we worked with the FDA in Washington to make sure that we could very safely proceed with testing of that therapy initially in the lab and then ultimately in 2 different animal models. And then we opened a clinical trial that was allowed that boy to be enrolled with, of course, his parents' approval and for him to be dosed. And the result was spectacular. And in fact, he was released from the hospital recently as a happy, healthy child, gaining lots of weight and looking very chunky. So,
Yeah. No,
really,
it,
really exciting. Yeah.
it's so amazing. I don't think people necessarily grasp this timeline that we'll post with this is just mind-boggling. How you could, as you said, Jennifer, in about 6 months to go from the birth and sequencing through cell-specific cultures with the genome mutations, through multiple experimental models with non-human primates, even looking at off-target effects, through the multiple FDA reviews, and then dosing, cumulatively 3 dosing to save this baby's life. It really just amazing. Now that is a template, and before we go to this new center, I just wanted to also mention not just the timeline of compression, which is unimaginable, and the partnership that you've had at IGI with, I guess, Danaher to help manufacture, which is just another part of the story, but also the fact that you're not just even with CRISPR 1.0 as being used in approvals previously for sickle cell and beta thalassemia, but now we're talking about base editing in vivo in the body using an mRNA delivery. So maybe you could comment on that, Jennifer.
Yeah, very good point. So yeah, we used a version of CRISPR that was created by David Liu at the Broad Institute and published and available. And so it was possible to create that, again, targeted to the exact mutation that caused baby KJ's disease. And fortunately, there was also an off-the-shelf way to deliver it because we had access to lipid nanoparticles that were developed for other purposes, including vaccinations. And the type of disease that KJ suffered from is one that is treatable by editing cells in the liver, which is where the lipid nanoparticle naturally goes. So we were, there was definitely some serendipity here, but it was amazing how all of these pieces were available. We just had to pull them together to create this therapy.
Yeah, no, it is amazing. So that, I think, is a great substrate for starting a new center. And so maybe back to you, Priscilla, as to what your vision was when you're working with Jennifer and IGI to go through with this.
I think the thing that's incredibly exciting, you mentioned that at CZI, our mission is to cure, prevent, and manage all disease. And it's when we talked about this 10 years ago, it felt like this far off idea. But every day it seems closer and closer. And I think the part that's super exciting about this is the direct connection between the basic science that's happening in CRISPR and the molecular and sort of down to the nucleotide understanding of these mutations and the ability to correct them. And I think many of us our imaginations have included this possibility, but it's very exciting that it has happened with baby KJ and CHOP. And we need to be able to do the work to understand how we can treat more patients this way, how to understand the obstacles, unblock them, streamline the process, bring down the cost so that we better understand this pathway for treatment, as well as to sort of increasingly democratize access to this type of platform. And so our hope is to be able to do that, go from take the work and inspiration that IGI and the team at CHOP have done and continue to push forward and to look at more cases, look at more organ systems. We're going to be looking, in addition to the liver, at the bone marrow and the immune system, and to be able to really work through more of the steps so that we can bring this to more families and patients.
Yeah, well, it's pretty remarkable because here you have incurable, ultra rare diseases. If you can help these babies, just think of what this could do in a much broader context. I mean, there are a lot of common diseases have their roots with some of these very rare ones. So how do you see going forward, Jennifer, as to where UC Berkeley, Gladstone, UCSF, I'm envious of you all up there in Northern California, I have to say, we'll pull this off. How will you get the first similar case to KJ Muldoon going forward?
Right. Well, the IGI is a joint institute, as you probably know, Eric. So we were founded 10 years ago as a joint institute between UC Berkeley and UCSF, and now we have a third campus partner, UC Davis. and we have the Gladstone Institute. So we've got an extraordinary group of clinicians and researchers that are coming together for this project and this center to make it a success. And we are building a clinical team at UCSF. We have several extraordinary leaders, including Jennifer Puck and Chris Dvorak, and they are both going to be involved in identifying patients that could be enrolled in this program based on their diagnosis. And we will have a clinical advisory group that will help with that as well. So we'll be vetting patients probably right after we announce this. We're going to be looking to start enrolling people who might need this type of help.
Do you think it's possible to go any faster right now than the 6 months that it took for KJ?
I think it could be. And here's the reason. There's a very interesting possibility that because of the type of technology that we're talking about with CRISPR, which fundamentally, and you and I have talked about this previously on your other podcast, but we've talked about the fact that it's a programmable technology, and that means that we can change one aspect of it, one piece of it, which is a piece of a molecule called RNA that's able to direct CRISPR to the right sequence where we want to do editing, and not change anything else about it, right?
Right.
The CRISPR protein stays the same, the delivery vehicle stays the same, everything else stays the same. And so we're working right now with FDA to get a platform designation for CRISPR that
Mhm.
might allow streamlining of the testing process in some cases. So it'll obviously come down to the details of the disease, but we're hopeful that in the end it will be possible. And Priscilla and I have talked about this too, that as AI continues to advance and we get more and more information about rare diseases, we'll be able to predict accurately the effects of editing. And so in some cases in the future, it may be possible to streamline the testing process even further safely.
Yeah, and I also would note, as you both know well, this administration is really keen on genome editing, and they've had a joint announcement regarding their support. And in my discussions with the FDA commissioner, this is something they're very excited about. So the timing of the new center for pediatric CRISPR cures is aligned with the current administration, which is good to see. It's not always the case. Now, going back, Priscilla, to your point that not just for the liver, because delivery has been an issue, of course, and if we're going to try to get after a lot of these really rare diseases, it's going to go beyond the liver. So this is also an exciting new dimension of the center, as you said, to go after the bone marrow for hematopoietic cells, perhaps other organs as well.
I mean, with the expertise and feasibility, the immune system is going to be the next target. Jennifer Puck has been a pioneer in this work. She's the one who designed the newborn screen that will be the tool that picks up these patients as they are born. And I think the thing that's tremendous is the immune system, first of all, is active in many, many diseases, not just these cases of children born with partial or absence of immune systems. And the course right now that these babies are left with is complete isolation and then a very long and arduous course of a bone marrow transplant with high morbidity and mortality. And even if after the transplant, you have complications like graft versus host and immunosuppression. And so the idea of being able to very specifically and with less sort of the conditioning and morbidity and mortality of the treatment, being able to address this is incredible. And the implications for other diseases like blood cancers or other hematopoietic diseases, that's incredible. And that actually has an incredibly broad base of patients that can benefit from the learnings from these babies with severe combined immunodeficiencies.
Yeah, I think that goes back to a point earlier, maybe to amplify in that previous CRISPR generation, it required outside the body work, and it was extremely laborious and time consuming, and obviously added much more to the expense because of hospitalization time. This is different. This is basically doing this inside the affected patient's body. And that is so one of the biggest reasons why this is a big step forward and why we're so fortunate that your center is moving forward. Maybe before we wrap up, you might want to comment, Jennifer, on how you were able to bring in to build this platform, the manufacturing arm of it, because that seems to be yet another dimension that's helpful.
Indeed, yes. And we were, again, fortunate with timing because you mentioned briefly that the IGI had set up a program with the Danaher Corporation back in January of last year. We call it our Beacon Project, and it's focused on rare disease. And it's a really interesting kind of a unique partnership because Danaher is a manufacturing conglomerate. So they have companies that make molecules, they make proteins, they make RNA molecules, they make delivery molecules. And so they were excited to be involved with us because they want to be a provider of these types of therapies in the future. And they can see the future of CRISPR is very exciting. It's like an expanding, growing area. And so that agreement was in place already when the baby KJ case came to our attention. And so what we're hoping to do with Danaher is, again, work with them and their scientists to continue to ask, how can we reduce the cost of these therapies by reducing the cost of the molecules that are necessary, how to make them efficiently. It's very interesting. Fyodor Urnov has toured their plant in North Dakota recently. And he found, in talking to their engineers, there are a number of things that we can already see will be possible to do that are going to make the process of manufacturing these molecules faster and cheaper by a lot.
Wow.
And so it's a win-win for everybody. And so we're really excited to do that in the context of this new center.
Oh, that's phenomenal, because some of these disorders, you don't have that much time to work with before they could be brain or organ or vital tissue damage. So that's great to hear that. What you built here is, the significance of it can't be underemphasized, I'll say, because we have this, May report of baby KJ, which could have been a one-off, and it could have been years before we saw another cure of an ultra rare disorder. And what you're doing here is insurance against that. You're going to have many more cracks at this. And I think this is the excitement about having a new dedicated center. So just in closing, maybe some remarks from you, Priscilla?
I just want to emphasize one point that's really exciting as we talk about these ultra rare cases that sort of, they're often like 1 in a million. All these learnings actually help maximize the impact of lots of research across the sector that impacts actually everyone's health.
Right.
And so our learnings here from these patients that are sort of have very significant presentations that really can stand to benefit from any treatment is hopefully paving the way for many, many more of us to be able to live healthier, higher quality lives through basic science.
And over to you, Jennifer.
Couldn't agree more. It's a really interesting moment. I think we're at sort of an inflection point where, as I mentioned earlier, all the pieces are in place to do this kind of therapeutic. And we just need a team that will focus on doing it and pulling it together and also learning from that process so that, as Priscilla just said, we are ultimately able to use the same strategy for other diseases and potentially for diseases that affect lots of people. So it's exciting.
Yeah, for sure. Now, if I could just sum up, this is now a decade past the origination of your work of CRISPR, and how already at the first decade culminated in sickle cell disease treatment and beta thal. Now we're into the second decade of CRISPR, and look what we've seen. Something I didn't think would be it was unimaginable until it actually happened and was reported just a little over a month ago. Now, going back to Priscilla's point, we're talking about thousands of different rare Mendelian genomic disorders, thousands of them, and if you add them all up of rare diseases, we're talking about hundreds of millions of people affected around the world. So this is a foray into something much bigger, no less the fact that some of these rare mutations are shared by common diseases and approaches. So this is really big stuff. Congratulations to both of you and your organizations, the Innovative Genomics Institute and the Chan Zuckerberg Initiative, for taking this on. We'll be following it with a very deep interest. Thank you.